So welcome and good evening. Thanks to all of you for tuning in this evening. I'm going to make two short announcements before introducing Dr. Tribley here. First announcement is that his presentation, his lecture will be recorded tonight, and we're going to post that on the Sheridan College lecture website where you found the URL link for this webinar. So look there tomorrow if you wanted to share that with folks who couldn't make it this evening. And then secondly, if you do have questions, we'll have kind of a Q&A session at the end of Dr. Tribley's lecture. You can shoot us those questions. There's a Q&A box at the bottom, hopefully, of your screen. You can send those questions, and then we'll have a look at them at the end of the lecture. So my name is Scott Newbold. I'm a member of the biology faculty at Sheridan College, and I help coordinate the Sheridan College Museum of Discovery lecture series. The lecture series has brought local and regional scientists to Sheridan for nearly 2 decades, and we're grateful for the long-standing support we've received from the Sheridan College Foundation and from the Life Science Department at the college. Tonight's lecture feels unique because we not only have the opportunity to gain some insights about the virus that has affected all of us over the last 9 weeks or more, but we may get a small glimpse into the mind and passions of our new college president, Dr. Walter Tribley. It's my pleasure to introduce Dr. Tribley tonight, not as the world's expert on coronaviruses, or even as a virologist, which he emphatically assures me he is not, but as a molecular biologist, and importantly, a passionate educator in the discipline of biotechnology. I suspect that one of the pieces of his career that he's most proud of is his work as a traveling biotech educator. With support from the National Science Foundation grant, Dr. Tribley developed, curated, and delivered traveling workshops to help faculty and graduate students discover a deeper understanding of the conceptual underpinnings and tools of molecular biology. Dr. Tribley earned a bachelor's degree in secondary science education and a bachelor's and a master's degree in biology from the University of Idaho. He earned his Ph.D. in biochemistry from Washington State University before joining us as president of the Northern Wyoming Community College District. In July 2019, Dr. Tribley served as the superintendent and president of the Monterey Peninsula Community College District from 2012 to 2019. Dr. Tribley has dedicated his career to the mission of community colleges, where student success is celebrated, employees are honored, and communities are empowered. The title for this evening's lecture is Emerging Infectious Diseases. Please welcome Dr. Walt Tribley.
Thank you very much, Dr. Newbold. It's just a real pleasure to have been invited by you and your colleagues in this distinguished lecture series. I want to reiterate to my audience how grateful I am for you to tune in. Tonight's lecture is different from the manner in which I certainly would like to teach in active learning style. Lectures are trying to take complicated subjects and distill them into manageable context to help you extend further. Tonight's subjects are a little ambitious. We're going to take on the subject of emerging infectious disease. And really, I'm just going to touch on a few of the concepts that hit us hit us clearly in the popular press and are hitting us now. The idea for this is to target primarily our students that are Zooming in tonight and everybody else. So I'm targeting this as a non-majors presentation. For our students that I know are on board, I will be using some of the terms and the concepts that I know that our talented faculty are using in the classrooms. And I want to add just a few comments to that overly generous introduction that Dr. Newbold gave, that truly education to me, and I think I've chosen the right career, education to me is the most important thing we do for each other, because we empower each other. And I learn from my colleagues every day. I learn from my cabinet members, I learn from students, I learn from my trustees, I learn from everybody. I've been blessed to have a mother who was very much engaged in teaching, who was also a nurse and worked for a while in the library. So that being said, we're going to go ahead and get started. There will be time for questions and answers at the end. I'll probably ask Dr. Newbold to come back in just to maybe help me if I'm stumbling through the questions and not seeing enough of them. So let's go ahead and get started. I'm going to share my screen. There we go. Okay. One of the blessings in disguise here of this COVID-19 has been that I now have to use the technology that I have been asking our faculty to move overnight to, and they've done such an admirable job. And I'm just very humbled by the work they've done so quickly and so well. So, all right, emerging infectious disease. You ever wonder where these new diseases come from? I mean, we didn't have this disease, now we have that. So the subjects tonight, we're gonna touch on the idea of how these things arise, if you will, or how they become apparent in populations. These subjects will involve concepts like mutation. They'll involve how these diseases evade being destroyed. And when it comes to bacterial resistance to antibiotics, how we human beings have created an evolutionary selection pressure and actually have helped select for resistant bacteria. So let's go ahead and move on. The common cold, there was a cheeky tagline on my title that was on the billboard out there in Coffeen Avenue at Sheridan College. We, of course, have two colleges in our district, one in Gillette, one in Sheridan, and we have a center in Johnson County in Buffalo. Here's this disgusting picture that I want to remind everybody that we need to, cough into our arms and wash our hands, etc. But it reminds us that droplets from humans are everywhere. We've all caught the common cold. I have this title that says common colds are not common because they're caused by at least 100 different agents. One of the agents that causes a cold is coronavirus. Coronavirus is a family of viruses. So in our biology, students know about families and the different taxonomies that we teach in biology classes. A family is a pretty large group. Coronaviruses, according to the CDC, account for approximately 20% of the common colds in the human population. Others are caused by rhinoviruses, up to 40%, and other causative agents. They've been around since we have, and we do not have a vaccine, and we don't have a cure. When we go to the store, we get remedies. We get things to treat our symptoms, help us sleep, clear our nose. Some of those things actually help the virus do a better job at harming us. But they stem our reactions, our inflammation, our runny nose, or fluids that we're producing to push out bad things out our various openings. One of the things I am not a medical doctor, but very often in our community college classes and in our graduate and upper division undergraduate courses, we do speak to medical applications as these are very important to obviously all people, and they make these examples relevant. Have the sun coming in there, reflecting off my full head of hair and making it hard to see. All right, so not your common cold, that's for sure, but coronaviruses that cause the common cold tend to infect the upper respiratory tract, our nose, our throat, and the upper part of our respiratory system. Whereas COVID-19, that disease can target lower in our respiratory tract and other places. All right, so we started with the virus, and I wanted to, here's a quote. I don't know if it's a quote, but I think you've heard it. If you've been to the doctor and you're showing up with all kinds of, maybe you think flu symptoms, you got a fever, you got runny nose, and you feel horrible, and you hear it's because your mucus is clear. It's not yellow or green. Yellow would be a real signal for a certain one. It's clear. You're not presenting like you have a bacterial infection, and you hear from that fabulous doctor who's doing the right thing. It's a viral infection, probably say something like most likely. So we are not treating it with an antibiotic, thank goodness. Not too many decades ago, you might have gotten an antibiotic. So let's start here with that familiar thing to all of us. Let's build from there. You see this diagram, and many of you know what these terms mean, and many of you don't. So the big division, in my view, of living things that have cells, living things that have cells, a virus doesn't have cells, right? Living things that are made of a cell or many cells are divided into these two gigantic groups. Those things that have a nucleus, they are eukaryotic cells. They have a true karyon. They have a true nucleus. And here it is in this diagram, the nucleus. Over here, these are cells, they're prokaryotic, that are thought to have evolved before prokaryon. In this case, the word karyon refers to nucleus and not the stuff that, not the word that begins with a C. That's the stuff that, you know, that vultures feed on. All right, so here we have two divisions. Most of the antibiotics that we are familiar with target the differences in how eukaryotic cells are made. We are eukaryotic organisms. We have nuclei in most of our cells, certainly not our red blood cells. They're just red bags of hemoglobin. And there's our skeletal muscle cells have more than one nucleus. There's always these exceptions. And over here, we see a bacterial cell. This one's a rod-shaped bacterium. It has a flagella, so it can swim around, and that stuff is DNA. In our cells, the DNA is in a nucleus. So they have a lot of parts, and you can see their names. I want to focus here on ribosomes. See, they have ribosomes. Oh, wait. We have ribosomes in our eukaryotic cells. I keep moving my screen because I don't have a way to block the sun. I don't have shades in my beautiful office. It's wonderful. It's temporary office. It's great. All right. Even though both cell types have ribosomes, they are structurally different. They are analogous in their function, and they both carry out protein synthesis. They make proteins. All right. There's a number of other differences between these two cells. Look at this really thick cell wall, and here's this wimpy plasma membrane. So you'll see later in our talk that some of the antibiotics target the cell wall because that drug can stop something that we don't have. That's a way for the drug to be specific, hit the target organism, kill it, and have less effects on the host, on us. So this is a theme of our talk. It's the way that antibiotics target the metabolic and molecular biology, the differences in the molecular biology between bacteria and between human beings. It's also going to underscore in this brief 50 minutes why we don't prescribe antibiotics to viruses. And I'll say now verbally that viruses hijack our cellular machinery to a great extent. Now I'm talking about, you know, there's lots of different viruses. Coronavirus comes into our cells. You'll see a very complicated diagram. Comes into our cells and takes over our machinery. So we can't give ourselves a drug that's going to knock out and destroy our ability to make proteins, which are needed in many trillions of cells. And we couldn't do that. We would kill ourselves. So therein lies the challenge, the lack of cure. That's one example of the challenge. All right, here we go, dear students. These are both eukaryotic cells. Let's start on the left. We're talking now about molecular biology to an extent. We're talking about the way genes give rise to things in the cell, primarily proteins. So in order for a cell to be something specific, you know, you and I were first a gleam in our biological parents' eyes, and then we became a cell. One from our biological father, one from our biological mother. We became a single cell. Most human beings would have 46 chromosomes in that cell. There are a few genetic diseases that have more chromosomes, but most of us have 46 chromosomes. That cell divides into 2 to the 4, 4 to 8, 8, 16, and 32, you get it, 164, 128, etc. What makes the cells different is differential gene expression. We have brain cells, they don't look like skin cells. They have the same set of chromosomes. So some genes are turned on that are the same in all of our cells. Those genes would be things like the genes that are needed to get energy out of sugar. You need energy out of sugar in your brain. You need energy out of sugar in your muscles. You need energy out of sugar in your skin, et cetera. But then you have certain genes that are selectively expressed and not expressed. So over here, the DNA is where we have genes. The process of transcription is a word that describes going from one oral language to be transcribed into written language, but we're still talking English. I'm talking English, believe it or not. And many of you might be writing and transcribing, but it's still English. So we're going from the letters, and these are terms that we use. Some of these terms, like transcription and translation, had their first role in describing language for us. So the language of DNA is transcribed into the language of RNA. They're very similar. The letters that make up the language are very similar. But then that RNA leaves the nucleus, comes out here, a ribosome reads that message and translates a nucleotide language, an RNA language, and translates it into a protein language. Over here to the right, we can see that this ribbon-like thing is supposed to symbolize the RNA molecule. And these letters down here are the actual subcomponents of that broader molecule. You biology students know these are the monomers that make up the polymer, right? The single individual units, things like uracil, guanine, cytosine, adenine. All right. So ribosomes then translate the message into proteins. That's like going from English to French. It's a translation. So these are the basic functions that we're going to focus on tonight. What science lecture wouldn't be complete without a black and white picture of a couple of some old dudes that did something really great, and these folks did. Here, this guy here is Alexander Fleming, and he discovered serendipitously, over here on the left-hand screen is an actual picture of the Petri dish that has bacterial colonies growing on it. These are, for our microbiology students, these are gram-positive bacteria growing here in colonies. That means they have really thick cell walls. And this is penicillium. This is a mold. And it is a mold that's secreting something in the medium here. And the medium is not solid, it's semi-solid. It's like a hard gel. And things can diffuse through it, like small molecules can diffuse through it. So he noted that something is inhibiting the growth of the bacteria. And this was it was a new discovery, certainly, of that molecule. But as a bacteriologist, then and today, they are very well schooled in the idea that things that live near each other in the soil or in our bodies are constantly competing with each other, constantly. We have bacteria on us and in us that make up our normal flora. And one of the things they're doing is competing for the nutrients that these pathogens want also. That's part of the equation. So Fleming discovered it, but he had really this is a really good paradigm for how academic research is so very important then and today. He discovered that. We wouldn't have penicillin and antibiotics, certainly not at the rate we did, if it weren't for that seminal discovery, if he weren't doing that research on things that are tiny and you couldn't see without a microscope and very esoteric. If he didn't do that research, if it wasn't funded, if it wasn't a university or a college that thought it was important, we wouldn't have this discovery. But he needed to link up with people that had production mindset, take an invention and now scale it so it could actually be useful for something, right? And that today is done in our biotech industry. Takes seminal ideas from academia and figures out which of those ideas can actually be a product. And then big pharma, Eli Lilly, Merck, Pfizer, those companies are really good at getting it out there and getting it to the people on large scale. So Fleming discovered it. Flory and Chain, these two folks were two of the people involved in producing larger quantities. There's one that did not get the Nobel Prize, last name Heatley. He was very instrumental, chose a different strain of mold that produced it at a greater amount than this one did. And he used the power of mutation. He shot it with X-rays to get that mold to produce 1,000 times more of penicillin G than the original mold. So that's a side note there, but I want us to see how academic research and community partners, these folks here were in academia as well, but they were very production minded. They were chemically minded, and they knew how to purify things in larger amounts. And that was something that Dr. Fleming did not, wasn't part of his skill set. And the three of them were recognized with the Nobel Prize. And Mr. Heatley got the first honorary doctorate from Oxford in 800 years, which is pretty good. All right, look out world, here comes penicillin G. And I am old enough, as some of you are too, I saw the participant list, to actually get one of those shots in the posterior, because that's the way it was delivered. Certainly antibiotics these days, some go in that way, and some are oral, and others are applied in different mechanisms. I just thought the picture was wonderful. One of the first administrations. Penicillin showed up being clinically available in the 1940s. 18% of the deaths in World War I came from pneumonia, from a gram-positive organism causing pneumonia. And in 1940s and World War II, we used these wars to try to magnify the horrors that we were trying to solve. Dr. Fleming was a doctor on the Western Front in World War I. Can you imagine how helpless and how many cases he saw of people dying from the infections of the wounds? Fixing the injury was bad enough. Protecting the patient from the infection had been the scourge of our human population, and it is today. So these are the things I feel that I project that must be having certainly motivated him and his colleagues. So here's an example of how our microbiology students sometimes are brought into this world. Our skilled microbiology teachers teach them how to create a lawn. That yellow background, that dark yellow background, if you follow my cursor, is a lawn of a bacterium. I'm making this up, but let's say it's Streptococcus, another gram-positive organism. And then each of those smaller white circles is really a filter. It's made of cellulose. It's a disc. Some of them are made of nylon. It's a disc, a small fabricated piece of material that has been saturated with, impregnated with, if you will, with different antibiotics. And this one doesn't have the antibiotic names on the slide. I wanted you to see the example. So our students in microbiology do this, and they spread the lawn out. You can't see the lawn like this. You're starting it out. It's all clear. And then at the same time, they use a dispenser to put antibiotics on there. They come back a certain period of time later, usually the next lab period, but they would start to see that the next day if they were there every day. And they look at the zone of inhibition or not. So let's go look at this one. Wow, that's got a nice zone of inhibition. This one has a nice zone of inhibition. Very much smaller, much smaller. Which antibiotic do you think is working best against this organism? Which one's not working at all? And these are the types of things we teach our students. And very importantly, our pre-nursing and pre-med students. This sometimes in a microbiology lab is among the first time. Now I know we do this in other venues. We do this in welding, right? We do this in machine tool and in diesel. They're around dangerous things. In a micro lab, you're around things that can kill us. We're around fire. We're around fire next to alcohol. We're around Staph aureus, Streptococcus, all kinds of pathogens, spore-forming bacillus, and they have to follow very strict, very careful sterile procedures. That is part of their affective domain growth, not just their cognitive brain. They have to have courage. They have to have confidence, and we're helping them build that. You, my faculty, our faculty are helping them build it. So anyway, that's the way we can determine. Nowadays, we have much faster ways of determining which organism is causing an infection, all kinds of rapid genetic tools. But I wanted to put this here because we're talking about resistance. This is a very complicated slide, the kind of slide that if I were observing, helping a faculty member, and my gosh, that was a pretty busy slide. You went through it pretty quickly, you read all the words. Our faculty don't do that. But I'm going to use this to show that this cell here is, this fictitious bacterial prokaryotic. Bacteria are prokaryotic. Should have mentioned that. It has a thick cell wall. Our cells don't even have cell walls. And it has a cell membrane like we have, but we don't have cell walls. So some antibiotics like penicillin G make it impossible for cells to actually make a cell wall. So they can't divide and replicate if you can't make a cell wall. They will break down and die. They will lyse. So penicillin G was one of these here, one of these types of antibiotics. It was a beta-lactamase. Over here, more cell wall inhibitors. There are some drugs that inhibit DNA replication. Our cells do DNA replication, but bacteria have enough differences in their mechanisms that you can target a drug there. Our cells do transcription, so do theirs, but differences in the molecular biology, differences in the enzymes, overall function the same, but very specific differences. Here, this diagram focuses on ribosomes. These two circles that says 50S, and on the bottom, I don't want you to look at it horizontally, look at it up and down. A 50S yellow thing and a 30S orange thing, and there's a bunch of them on this line. This line is messenger RNA. You saw that in a previous slide. It's an RNA transcript of a DNA message. It's an RNA message. It is transcribed, right? So it's very similar, but it's in RNA language. And here it is being translated into proteins, and all kinds of drugs target that process, especially some of our broad spectrum antibiotics. Penicillins are narrow spectrum, meaning they attack a certain narrow, very important, very toxic, very pathogenic, but a narrow band of bacteria. You don't know what's causing the infection. Tetracycline is an example of one that spans a range of gram-negative thin cell walls, gram-positive thick cell walls. So my point here is all of these antibiotics target various parts of the molecular biology or the metabolism of a prokaryotic cell, a bacterial cell, in ways that don't harm us. However, our liver typically and we have to metabolize these drugs. They have other effects. And thankfully, we have a talented medical community that keeps us on top of the game. Before I move on here to coronavirus, I wanted to stop and say, it's time for me I'm going to try this. I hope I don't lose you all. I want to go back and stop sharing for a second. All right. We are into our lecture about a little less than halfway. And I'm only going to do this orally because I think the basic principles are going to be very straightforward, and there's lots of details and lots of science to back this up. I'd like you to imagine a fictitious person right now. And we're going to call this person patient 0 or patient 1, the OG, the original gangster patient, who presents with an infection to a clinician. And this person is, patient 1, right? If we're going back, it's a fictitious story that is a true scenario. This person has been infected by a staphylococcus. He has a staph infection. And little does anybody know that in the population of bacteria that are causing that infection, there is a tiny number. Some of the folks watching this would know I'm creating quite a bit of fantasy. Let's say 2% of the total number of individual bacteria in that population causing the infection have mutated. It happens. Mutations happen, and they lead to traits, phenotypes, things that species do. So bacteria mutate at a very rapid rate, simply on our time scale, because they divide so quick. There's so many divisions, so many DNA replications. They live out in hostile environments with UV light hitting them. There's all kinds of ways that bacteria mutate. 2% of the 2% of that population in this example is resistant to the drug that's being prescribed by a fictitious, in his fictitious example. So now when the drug is prescribed, those 2% are not touched by it. It's like, you know, it's no big, they don't have any effect. But our immune system, in this example, it's a healthy individual with a fully intact immune system. And I'm going to stop here and say, I hear, if you stop taking your antibiotics, you will create, that word, you will create resistant bacteria. And I'm want us to take pause here for a second. In this example, 2% of those bacteria are naturally, they mutate, they have this trait, haven't had to use it, but they have it. That happens very, very rarely. So it's there. I mean, in bacteria, they mutate frequently, but it's traits evolving for yet there is not this challenge, this environmental challenge. So the patient takes their drugs, and at the same time, the immune system's attacking these. The immune system is attacking 100% of them equally. The ones on the outside near the blood supply are getting hammered first. The ones inside this population are cozy for a while. But the resistant bacteria are getting eaten, getting killed just as fast as the ones that are not resistant. Everything's going good. And that drug that the clinician prescribed was good because it's taking care, it's helping our bodies. It's an ally with our immune system, helping our bodies to fight that infection. And together, it's a 1-2 punch. And 98% of those bacteria are getting hammered from at least 2 fronts. Now, what's happening elsewhere nearby this infection are all, I'm going to say, am I going to say this? I'm going to say all the good. They might not be good. They're just there. But the good bacteria, the normal flora that are hanging out, they're competing for space. Space is places to attach to us and then send digestive enzymes into our sensitive tissues, our very sensitive tissues. So space is important. If you're trying to put your house on top of a crowded city, you have no space. But all of a sudden the antibiotic starts taking care of, and it starts killing, and it starts making more space and more nutrients are available. But still, that drug is doing a good job and we're on a great track. And then guess what? The guy starts feeling better and says, I really want to stop taking this drug. I know it's a pain in the butt. I'm not, excuse me. It's a pain. And I'm going to stop taking this drug. I'm just going to forget. So now what's happening is all the bacteria come racing back. They divide so quickly. One bacteria today in a logarithmic exponential growth, the number you have tomorrow is a 1 with 24 zeros after it. And I have to ask our math faculty, what is a number with 1 and 24 zeros after it? It's a lot of bacteria. So they stop taking the drug. Now that 2% starts dividing. And when we come back to the clinician, we're no longer looking at something that we can manage. That drug isn't going to stop it because now we don't have 2% that our immune system will take care of. We now have 98% that are resistant, 2% that might not be, and our immune system's overwhelmed, and we might have to scrape the bacteria out and pour topical stuff on it and other disgusting things. I hope you have already eaten. So with that being said, we're going to move on, and I'm going to go back to sharing our document. Okay. That wasn't so good. Let me catch up. There we are. Coronavirus disease 2019. That's the disease, COVID-19. It is caused by severe acute respiratory syndrome, coronavirus-2, different from SARS-CoV, the first one, -1. I want to mention here that we had an episode of SARS in our human population, not SARS-CoV-2, but the original one. It had a higher percent of the cases I read were much more serious. It's hard to get more serious than dying, but I'm saying a greater percent of the cases were more serious. the infection, apparently, I read, presented itself more seriously quickly. Whereas SARS-2, first of all, binds to its receptor over here in the picture. It binds the receptor. ACE2 receptors, part of our blood pressure control mechanisms. They're sneaky, these viruses, gets into really important cells that way. But it binds more tightly. But one of the tragic things here that our medical professionals are dealing with, I read, is that many people don't have severe symptoms, and they are infectious to others while they're not feeling it. So that makes it very hard. Whereas in the initial SARS infection, they were easier to identify, easier to social distance, and therefore easier for us to move through. That's one difference, how this is not the common cold. This hits the lower respiratory tract and other places. Wherever there's an ACE2 receptor could be hit. I read that there are symptoms that are very difficult to explain, immune systems being infected in some small children. There are blood clots that are forming. If it's impacting the heart, the folks that understand cardiology better than me, we know that when our heart has trouble, we have to sometimes take blood thinners because our blood is not, if our heart's not functioning strongly, the blood will pool and has a tendency to clot. So blood clots it makes sense to me as a lay person that this would be part of the issue. Anyway, here you have a very complicated diagram. The point I've already made. Virus here attaches to a cell specifically. There is, I didn't make that point before, but I'm making it now. And then it's brought into the cell, and then it goes through a number of molecular biology steps that it primarily hijacks our human cellular machinery to replicate, kill the cell, make more viruses to infect other cells. So that's the main point. We can't give this cell an antibiotic right now. We can't stop its ribosomes. You remember I made a point about, and that yellow thing and that orange thing, and one's 50S, one's 30S. I didn't talk about what S means. These are the sizes of those subunits of ribosomes. They're different. Those ribosomes have the same function in bacteria, different from ours. Virus is using our ribosomes very efficiently. Comes in and it's off to the races, translations, viruses are made. Obviously, this is a very complex virus, this life cycle here. It's simple to a molecular biologist because of the efficiency, and it's hard to maybe for the layperson hear me say that. Viruses are really highly efficient in how they pack their genetic material. And I'll speak maybe a little bit to that with HIV in a second. So there is no cure for this, just like there's no cure for the common cold. We have to help our immune systems fight it. So people who don't have strong immune systems, people who are already weak with their cardiovascular health, people that don't heal fast, diabetics, others, and very often diabetes goes with cardiovascular health at a certain age. So these folks are particularly at risk, of course, and perhaps many, many others, people taking immunosuppressants for a variety of reasons. I want to remind, this was to remind us of how we have a cellular machinery, and these were our eukaryotic diagrams. I spoke to it orally, but these ribosomes are being hijacked by the virus. So you can't target most of those other places. You can't target a cell wall, you can't target DNA synthesis, you can't target many things here. They're hijacking our machinery. So that makes trying to combat a virus very, very hard. I go back to the times we've heard our talented and very smart clinicians who say, I think it's a virus. I'm almost sure it is. We have clear mucus, this, that. It's presenting that way. I'm not going to prescribe an antibiotic. I mean, that could actually do more harm than good. And certainly on a global scale and evolutionarily speaking, it does do more harm than good. Human immunodeficiency virus, HIV, and again, in the span of my career, and actually when I was engaged in doing some of the work that Scott talked about at the University of Florida with my colleagues, I was a member of a team that did biotech workshops, and I worked with some very talented, very excellent teachers. And we tailored workshops to the different audience. When we gave, and what we were teaching are molecular techniques. Yeah, late 80s, early 90s, we were teaching DNA sequencing and protein sequencing, how to make a monoclonal antibody, how to clone a gene, how to overexpress a protein, all that stuff. That was new. I mean, really powerful, important, smart, gigantic labs in the biological sciences did not know how to use molecular tools. Lots of thinking was turned on its ear. When I was teaching biology, there were 5 kingdoms. How many kingdoms are there now? That was turned on its ear because of molecular techniques. Anyway, in HIV, targets a cell in our immune system, a so-called helper cell. Couldn't be more diabolical in its target. That cell helps our immune system figure out invaders from non-invaders and what kind of response we're going to have. Antibody response, cellular response. How it's like it's the forward operations. It's the recon battalion. It's very important to our immune system. It enters those cells and it goes through this very complicated life cycle. It's called a retrovirus. It's another RNA-containing virus. Coronavirus contains an RNA genome, not a DNA genome. But this one does too, but it goes through some more steps. First, that RNA is reverse transcribed. Transcription is going from DNA to RNA. It goes from RNA to DNA right here. And then that DNA integrates into our genome and it gets replicated along with the cell's replication until some triggers gene expression, and then they're off to the races, and we get transcription and translation. This is wrong. I've chosen a diagram from the international, our www. This is not correct. We do not have translation to RNA. We have transcription to RNA. We have translation to protein. The idea here, though, the picture is nice. The RNA comes out of the nucleus and out into the cytoplasm. Ribosomes jump onto it, start protein synthesis, and then HIV proteinase, a protein that is coded for the HIV by the HIV genome, clips the certain proteins and makes them functional. The HIV cocktail of therapy targets HIV proteinase, and it targets reverse transcription. We don't do reverse transcription. None of our cells, as far as I know, at least 20 years ago when I was paying attention, none of our cells have reverse transcription going on, carried out by the enzyme reverse transcriptase. So that cocktail targets that. No vaccine. No vaccine. It changes too much. The mutation frequency on the proteins that are on the outside of this virus is too much, and our immune system can't even recognize the initial strain that infects us is not necessarily the strain that we'll find in a person that has acquired immunodeficiency syndrome, has the disease. So mutation also protects these viruses from immune response. Finally, the Staphylococcus aureus story. Staphylococcus means cluster of grapes. And here's a picture of some right there. It's stained, obviously. This is my second to last or so slide, and I just want to talk a little bit about evolution and selection pressure, et cetera. I did with that fictitious patient. Remember I talked about the guy, he stopped taking antibiotics. The antibiotics were a new agent in the environment. And they created selection pressure. And the organisms that survived, survival of the fittest, the organisms that were reproductively successful, they reproduced, were those 2% of organisms that had, luck of the draw, they had the genes for resistance to that drug. In bacteria have a form of bacterial sex, sex being defined as transfer of genetic information. There are pictures of so-called male bacteria transmitting DNA to a receiving bacterium. Genes for antibiotic resistance are often on those pieces of DNA that are transferred. Those things are called plasmids. I know many of our students have heard of plasmid. But I want to bring us back here. In 1945, Dr. Fleming, in his acceptance speech in the Nobel Committee, nearly pleaded to not overuse antibiotics, because this is a guy that was doing his research before we knew that DNA was the molecule of heredity. Think about that. But he understood, he saw the evidence in his daily work, or during the course of his work. It was very common to see microbes change their ability to survive, their ability to survive against competition, their ability to survive in different environments. So what we do when we overuse when we overuse antibiotics, not necessarily, we often in academics use this medical example because many of us have experienced it. The majority of this use were in things like growing lots of poultry. Imagine a poultry house back in the 70s with rows and rows and rows of chickens, the ones on the top defecating on the ones on the bottom. You lace their food with antibiotics and you can increase the density of your production, make more money, and create a product. Nobody knew that we were creating a world that had antibiotics in the environment, creating a selection pressure to enhance the success and survivability of resistant bacteria. Virtually none of the bacteria that cause infection today would be harmed at all by the penicillin G that was nearly 100% effective on gram-positive bacteria, staph, and strep back in the 1940s. So with that being said, we'll wrap it up here and we'll go to questions. I've highlighted a few. I'm a little bit early on our questions. That's probably good. We'll have a chance to do some, not necessarily discussion in this format, but some question answer. So I have highlighted certain aspects. You can read beyond if you'd like. But we've talked about how mutation drives genetic diversity. I haven't said that phrase, but in context told stories about how a mutation can give rise to a phenotype, that a trait, an appendage, an enzyme that cuts up an antibiotic, or a pump on the outside of a bacterium that when the drug comes in, it gets pumped out. True story. Enzymes that cut the antibiotic up. True story. All of these are possible. Mutation drives the genetic diversity. And then environmental factors. Number two, the environmental factors help select for these traits that then add to the survivability, which we biologists, at least when I used to be one, is really about being reproductively successful. That's what we mean by survival. It's not, that you have the big muscles or whatever. It's that you pass on your genes. That's survival of the fittest. Genes can be transferred. I gave the one example of how bacteria have sex. Viruses do the same thing. In both directions, there are some human cancers that are caused, they are certainly enhanced because some certain parts of viral genomes stay inside, when the virus leaves, stay inside the cell and cause a particular gene to be really overused. We would say overexpressed. Likewise, pieces of DNA come and go into viral genomes. It's obviously not a willy-nilly type of deal. Bacteria, same way. They can pick up genes from their hosts and they can deliver some genes. And I want to end here because, it's a college president and we're talking to you from our treasured Northern Wyoming Community College District. And we've talked a little bit about the importance of supporting research and supporting science, et cetera. But I want you to think about this, dear students in particular. Our faculty know this like the back of their hand. Do you ever wonder why in a college degree, let's say you're a student, you're going for a college degree at Harvard, here in the UC system, and you have to take general education requirements, but then you get to pick. They're actually trusting students to pick. The academy decides. The academy is our word for our faculty. That's why it's called academics. It's about that. So the Academy has decided that a well-rounded college-educated person with a liberal arts education has to have so many classes in this area, like in the sciences, so many in humanities, social science, etc. The student gets to pick. So all sciences, so if a student, so how could that be? The degree has to be common in some way. It's coming from our institution. We care about this stuff very deeply. You pick a course in physics, you pick a course in chemistry, geology, earth science, biology, you name it. They all should help us learn to think analytically, to think critically, to base our decisions on, we make our decisions based on conclusions that come from really well done experiments. We call those controlled experiments, right? But now I'm just talking about it more broadly because I want to leave you with where we are today. We have all kinds of forces trying to influence our decisions. We have political forces, we have economic forces, and we have scientific forces. And our leaders, like our dear governor and others, have to make decisions taking all of this into account. And I certainly appreciated when our governor said, we're going to be making decisions based on the data and not on a date. And we're not going to allow politics to influence us. That, of course, sang to me, because as a science educator, we're trying to get people to analyze situations, make good decisions for themselves. So with that being said, I am very pleased and honored to be with you here tonight. And I would like to welcome Scott Bailey back. Scott Bailey, did I say Scott Bailey? That was an art faculty member back in the day at Wenatchee Valley College. Dr. Newbold, and we'll get to your questions. All right, Q&A. There you are. Hiya, Dr. Newbold. I'm sorry. Bailey,
no worries. Here we are. So, Dr. Tribley, can you see the questions here, or you want me to read them out loud?
No, I can. So I'll read them out. I'll read them out loud. This one, I haven't read it yet, but I'm just going to take it as it comes. Good evening. Sorry it's a bit late, so maybe you already answered this question. Don't worry about that. Do you believe virus epidemics will become a common thing that humanity will face in the future permanently? Thank you in advance. This is coming from a business student. And my answer is yes. The answer is yes. And we are also evolving and changing. And we are also understanding more as we go. It's not, we are biological entities as well, right? We are adapting as well. So the nature of this talk tonight was to simply, was to get to this very great question that yes, that emerging infectious diseases are a thing that we have always had to work to help protect ourselves, and we will continue to need to do so in the future. And I'm sorry I don't have a more happier answer for that, but our tools and our techniques are also advancing. Our ability to detect these pathogens, our ability to create designer drugs that we can literally see fitting lock and key using our technology today. So I want you to have hope, but yes, the answer is simply it's going to be yes. How does a virus take over and make its own DNA? It's just because our immune system is weak. Yes and no. It survives because our immune system is weak. But viruses like the HIV, for example, HIV enters the cell with genetic information. You use DNA. We're going to get to DNA. It enters the cell, let's say the helper T cell, also called the CD4. It enters the cell in the immune system. And so far, that cell's not dead. It comes in with genetic information. It's in the form of RNA through a variety of a number of steps that RNA, it serves as a template to make viral DNA, and then that viral DNA literally gets inserted into our DNA, and our DNA that make up our chromosomes, right? It gets inserted. There's an enzyme called integrase that is a viral enzyme that it comes in with the virus, and it cuts and it helps integrate the viral genes into our genes. and it gets replicated along with, if that cell divides into two, the viral DNA goes into two, and those two into four. Now we have 4 cells with viral DNA. And then some trigger triggers those viral genes to then be transcribed and start producing the products that will lead to viruses that then kill the cell and are released into the bloodstream. So that's how it does. It has a mechanism to insert the DNA into our cells. Coronavirus works differently. Not all viruses work that way, but HIV does. Do you think then, this is in response to the first question, do you think then, in response to this trend, economies of the countries should localize to defend domestic economies? I'm actually glad that answer to my last question was yes, nice for my research. You know, to be honest, I do not understand. I am a non-major, and to your business terms here, Scott, do you know, I don't know what maybe the person asking the question could help me. I'm not understanding the concept of countries localizing economies. I need some help.
I'm unsure there as well.
See
if that person can clarify with another question, we can come back to that.
I do believe, as he's coming back, I do believe that as off of biology and into my administrator community development hat, I do believe that it's very important for us to invest in our local economies. There are people on this tonight that I know are out from different states. It's important for them to invest in their local economies because those dollars that are earned locally circle and they circle through our community with greater impact. So if a construction company is from this area and they're building something, and then they go out and get a haircut because they can do that now, and they one of 20 people in a restaurant or coming to college, they spend their dollars locally, and that helps. So maybe that's what you're referring to. But I do believe that this COVID thing has helped us understand the importance of not being too dependent on other economies for your basic needs. It's really a tough thing for me to say as a college president. The world is flat. We are all connected. We can't shelter, from the rest of the world. We have to engage with the rest of the world. And at the same time, we do have to be able, in this type of a crisis, be able to do for ourselves. And I do believe that we've seen that, certainly even in our local communities, making our own face masks and shields and making our own hand sanitizer, things like that. All right, Dr. Newbold, I am not seeing any other questions. Oh, there's 6 there. Am I not scrolling?
If you scroll down, yep.
Scrolling down. I'm a graduate of the Sheridan College Dental Hygiene Program and learned recently that dental hygienists are the #1 profession most vulnerable to COVID-19. How can we protect ourselves, and how will the students learn this? And she gives her name, Sheila Flynn, class of 1994. So I'm out of my element here. I do know that we are ordering enhanced personal protective equipment for the instruction of dental hygiene students. And we are not fully, we're not fully equipped to welcome all of our students back right now. We're anticipating that it's going to take a number of weeks to do that. So we just, certainly in a teaching environment and then out in the industry and working, we have to be very careful. Of course, yeah, because they have their hands and instruments in where lots of droplets and very close to those oral excretions, if you will. So we will just have to make do with the personal protective equipment and procedures and hygiene procedures and social distancing and waiting rooms, et cetera. I hope that was helpful. I'm listening from Western Montana, and I wonder how we can respond to those who express their belief in herd immunity in a way they can understand. Herd immunity. Dr. Newbold? All right, if this is coming from Suzanne, we still use antibiotics in agriculture. We still use them. my example was, you know, a real misuse because of the overcrowding in poultry situations. I mean, that's over the top. I think spacing out our animals, you know, social distancing, right? There isn't any social distancing in an overcrowded poultry house back in the 1970s. So I think that most of the operations that I have had, which isn't many, that I've had the opportunity to see, there's quite a bit of space for the animals, but I'm sure I'm not helping there, Dr. Newbold. I'm not understanding the herd immunity part. Oh,
I don't think I can help there either.
Okay.
And I think,
yeah, I see the clarification from our first questioner.
Yeah.
So in terms of localizing economies, meaning that there will be declining volume of trade on the global market for sure, and then he gives an opinion about moving us towards socialism. So meaning that there will be a declining volume of trade on a global market. Yeah, there has been, and so we will have to rely more on our local markets. I think your question makes that point. We're already seeing that now. We have to then figure out ways to re-engage globally. We will have to re-engage globally, and I think particularly in the United States, where so many of our raw materials and other parts of our economies come elsewhere at the same time. Particularly, I'm here. I appreciate the person coming from Western Montana. Here in Wyoming, we do a lot of mineral extraction. There's a lot of minerals that we can be extracting to use, and besides the ones that we already are now, that can be used in parts and in economies. But I'm certainly out of my element there. He makes a statement about shifting ideology towards socialism. I'm concerned also that it's more isolationism, which our country is no stranger to. We had sort of an isolationist view. It took Pearl Harbor for us to engage in World War II. And, you know, we have that in our idea. It's part of our independence, I believe. The isolationism thing. I can't comment on the socialism thought. I get a nice clarification from one of our faculty members. Herd immunity refers to the community having a high incidence of immunity of disease, 70 to 90% immune, which protects the others in the community of people. So that is true. I agree with that. I think that is a very helpful thing. I've run into that recently because my wife and I have welcomed a puppy into our family. And guess what? There are not many dogs in this area that have parvovirus. So there is an immunity because of the lack of incidence, and other members of the herd are protected that way. Thank you for that clarification. Dr. Newbold, I'm gonna...
Yeah, maybe we can wrap it up, and if people want to continue to provide questions, we can tackle those and post those along with a video recording of tonight's lecture.
Okay, I'm going to go ahead and end. It's 8:03. Thank you so much for your participation tonight. I greatly value that, and I think together we're going to get through it. I want to give a shout out to our great faculty and staff for all they are doing in response to this environmental challenge. We certainly are adapting and surviving. So thank you, and thank to our students for doing their part as well.